Okay, students. So good morning. Can you hear me, see me, and can you see my PPT? Right? You can see my PPT, right? See, you need to respond. Can you hear me, students? Am I audible?
Yes, sir.
Okay, thank you. So yes, students. So good morning. So the subject is, as we know, it is histotechnics, immunohistochemistry, molecular pathology. So and the subject code is 21 MSH 716, and currently you are in third semester of your masters in MLT. So the unit one. So if you coming to the unit one in your histotechnics, the unit one is all about enzyme histochemistry. So the idea of this particular topic is, so here we are trying to localize the enzymes present in the tissues. So here we are trying to visualize the functional enzymes within the tissue. Okay. Whereas in immunohistochemistry, we detected the antigens within the tissue. Okay. Antigens can be anything. Antigens means proteins. Okay. Any protein can be detected with the help of our immunohistochemistry. Whereas our enzymes will be detected by a specialized technique called a specialized branch called enzyme histochemistry. Okay, there is a statement, students. All enzymes are proteins. Okay, but not all proteins are enzymes. Okay, that means we can able to detect enzymes in immunohistochemistry, but not only enzymes, we will detect all types of proteins in immunohistochemistry. But if you want to specifically target enzymes, then we have this special branch. Okay. And what is the significance of studying these enzymes? What is the significance of understanding the role of enzymes or the presence of enzymes within the tissue? Okay. So as we know that enzymes are the primary substances that facilitate a faster metabolic reactions within the tissue. So all metabolic reactions, if you take vital metabolic pathways like glycolysis, gluconeogenesis, Krebs cycle, electron transport chain, shunt pathways, all these metabolic pathways can only be enhanced or they can be effectively metabolized only in the presence of enzymes. Enzymes plays a crucial role in the metabolism of various biomolecules. Okay, no matter of glycolysis or anything. Okay, all metabolic activities will happen only in the presence of enzymes. So understanding the functional activity of enzymes is very important in order to assess the functional status of the tissue. Okay, so and now these enzymes are specific in their locations. Okay, some enzymes are really specific to some organs. So let us take LDH, lactase dehydrogenase enzyme, isoenzymes. Isoenzymes means these are the subderivatives of the lactate dehydrogenase enzyme. Okay, the subsets of LDH can be isoenzymes. Usually LDH1 is prominently seen in heart. LDH2 is prominently seen in reticuloendothelial system. That means your phagocyte cells, your white blood cells in that reticuloendothelial system, we will see this LDH2. Whereas LDH3 is seen in lungs, LDH4 is seen in kidneys, and LDH5 is seen in liver and striated muscles. Okay, now if LDH1 concentration is less in heart, means that indicates there might be some problems with heart. The patient heart might prone to some ischemic heart attacks. Okay, so seeing the functional activity of enzymes within the tissues will have its own clinical significance. We can able to understand the functional status of that particular organ depending upon indirectly assessing the functional activity of the enzymes embedded within the tissue. Okay, so understanding enzymes is another important, it has its own significant clinical importance. Okay, so as I stated, enzymes are specific in their locations like LDH1 heart, 2 reticuloendothelial system, 3 in lungs, 4 in kidneys, 5 in striated muscle. Okay, so now we are trying to we are trying to locate the enzymes within the tissue in enzyme histochemistry. Okay, so this has many clinical applications. Okay, so the basic idea is to visualize the enzymes within the tissue. How we are going to visualize enzymes within the tissue is the idea of our enzyme histochemistry. So let us go to the syllabus. So in the syllabus, students, they have directly given us the topics. Okay. So in enzyme histochemistry, they said basic fixation, then cryo fixation. Then they directly went to the demonstration of various enzymes. Okay. Like demonstration of phosphatases. It can be acid phosphatase, alkaline phosphatase, or adenosine triphosphatases. Then dehydrogenases, demonstration of dehydrogenase in the tissue, demonstration of oxidases, esterases, and tyrosinases within the tissues. Okay. So before going for individual demonstration of individual enzymes, now first let us understand few basics about enzymes. What are enzymes? What is the classification of enzymes? What are the properties and what are the properties of enzymes? What are the principles? On what principle the enzymes are going on? Or what are the functional? What are the reactions of enzymes? What are the simpler terms of enzymes? Okay, there are some terms like substrate, primary product derivation, inhibitors, and what is the role of various types of factors like pH, its enzyme concentration, substrate concentration, even the status of the fixation of the tissue. So all these general fundamentals need to be studied before studying or before going for individual enzyme demonstrations. So now in today's lecture, I will discuss in detail about the basics in enzyme histochemistry. These fundamentals are very important in order to understand individual test. Okay. So in today's lecture, let us discuss about basics in enzyme immunohistochemistry. First, I will discuss about basics of enzymes. Then I will discuss about basics of enzyme histochemistry. Okay. And also keep in mind, students, in my lecture, I'm not going to teach you entire enzymology, that is biochemistry. Okay. I'm not going to teach you in-depth specific details of enzymology. Rather than we will have a surface overview, a basic overview of what are enzymes. Okay. So don't expect that I will go up to the level of biochemistry in order to make you understand of enzymes. Okay. I will give just a brief overview of these enzymes. Okay. So let us see what are these enzymes, what are their products, and what are the terms we are using in the enzyme histochemistry and enzymes as overall. So yeah. So coming to the basics about enzymes. Okay. So now students, so enzymes means enzymes are just like antigen antibody reactions. Okay. A specific enzyme can only react with a specific substrate. If I take a proteolytic enzyme will only act on proteins. It won't act on carbohydrates, lipids, or any other things. Okay. So enzymes are specific in their reaction. So an enzyme will bind with its substrate. Okay. It's a specific substrate. Upon binding, it will temporarily make enzyme substrate complex. Okay. An enzyme binded with a substrate and it made enzyme substrate complex. Okay. Now this enzyme substrate complex will make a product. It will convert the substrate to a product. Okay. So the idea is conversion of substrate to another product is the idea of enzyme. Enzyme will convert substrate to product. Okay. After converting the product, this enzyme can be reutilizable. This enzyme can be reutilized by the body. Enzyme reactions are reversible reactions. An enzyme will bind with substrate, it will convert substrate to product, and this enzyme will go for next to substrate. So enzyme can do these reversible reactions. Okay. So this is the basic terminology of enzyme activity. Okay. Enzyme will bind with substrate. It will temporarily make an enzyme substrate complex and it will convert substrate to a product. Okay. So this is simplest definition of enzymes, and enzymes are called as biocatalyzers. That means enzymes will increase the rate of chemical reactions. Okay. They will either increase the rate of chemical reaction or they can also decrease the rate of chemical reaction. That depends. Okay. Usually enzymes will enhance the speed of reactions. Okay. The process of speeding up any chemical reaction is called catalysis. Okay. So catalyzers. So enzymes are a very good biocatalyzers. Okay. They will speed up the chemical reactions. So yeah. So what is a catalyst? As I stated. Okay. As I stated. So a catalyst is a substance that changes the rate of chemical reaction. Either it will speed up the chemical reaction or it will slow down the chemical reaction. Okay. After reaction, a catalyst is released to combine again with another chemicals. Okay. After participating in the enzyme reactions, now this enzyme will go for next substrate. Okay. And what is a substrate? So substrate is usually either an organic or ionic component that will get affected by enzyme. So substrate will be converted to product. So that is called a substrate. Okay. So now you know what is an enzyme and what is a substrate. Okay. Now the properties of this enzymes is very, very, very important. The properties and factors influencing enzymal activity. So now let us see basic properties of enzymes. So enzymes are biocatalysts. That means they will increase the chemical reactions. Okay, they will increase this chemical reaction by lowering the energy input. Okay, as I stated, we are not going to study this much depth enzymal activity. Okay, there is a graph. In that graph, you can see how the enzyme speeding up a reaction. It will speed up the reaction by decreasing the energy input to any particular substrate. That is there is one concept. Okay, you just remember that enzymes will decrease energy input so that activation energy will be increased, and that is how they will increase the speed of chemical reactions. And all enzymes are proteins. Keep in mind, all enzymes are proteins, but not all proteins are enzymes. All enzymes are proteins, but not all proteins are enzymes. A very important statement. Okay, and enzymes remain unchanged after a reaction. So after participating in any chemical reaction, enzymes won't get changed after chemical reaction. Okay, they are unchanged after chemical reaction, and they can be reutilized for the next substrate. So they are unchanged in the chemical reaction. Then enzymes are specific to a substrate. Okay, each enzyme can only bind with its specific substrate. Okay, so enzyme is just like an antigen antibody. An antibody only binds with a specific antigen. Similarly, an enzyme will only bind with a specific substrate. Okay, this is called lock and key mechanism. Just like our key, one key can only fit into one lock. The way enzymes also only fit to a specific substrate. Okay, and enzymes are reversible chemical reactions. Okay, enzymes are reversible chemical reactions, and enzymes can be easily denatured. The activity of enzymes can be easily deactivated either by subjecting it to various temperatures or changing the pH. If you don't want the enzyme to work, then better change the pH or change the temperature. By altering these factors, the enzymal activity may be decreased. Okay, that is why understanding the factors influencing the enzymal activity is also important. So what are the factors that contributing to the normal functional activity of the enzyme? So one is concentration of enzymes. The higher the concentration of enzymes, the higher the rate of reaction. Then concentration of substrate. Optimum substrate concentration is required for optimal maximum activity of enzymes. If the substrate concentration increased, means the enzymal activity will decrease. If concentration of substrate decrease, then enzymal activity will increase. So concentration of substrate will affect the enzymal activity. Then concentration of products. Now substrate has been converted to product, and this product cannot be binded with the enzyme. So the higher the products, the lesser the enzymal activity. So concentration of products will influence the activity of enzymes. Then temperature. Higher temperatures leads to denaturation of enzymes, results in destruction of enzymes. So temperature will play an important role. And also at specific optimal high temperatures, the rate of chemical reaction will increase. Okay, at one particular point, enzyme sometimes if the optimal temperature is 5, but if we increase optimal temperature to 6, the enzymal activity will increase only up to a limit, only up to 6. If it's going beyond 6 or beyond 8 or 9, then enzymal activity will be stopped. Okay, so yes, so temperature plays a vital role. Higher the temperatures, the better the enzymal activity. Lower the temperatures, low enzymal activity. Okay, so low temperature, low enzymal activity. High temperature, high enzymal activity. Then pH. So every enzyme has its own pH, specific pH range for its normal activity. Higher pH or lesser pH may result in inhibition of enzymal activity. Okay, so pH also plays a crucial role. Then activators. So sometimes along with the substrate, we can able to accelerate the enzymal activity. Okay, and such enzyme such things are called activators. They are also called coenzymes or cofactors. So coenzymes and cofactors will further facilitate the higher activity of the enzymes. Okay, so that is the role of activators and co-activators. Then inhibitors, as the name states, Inhibitors will inhibit the enzymal activity. Then time for the chemical reaction. If you incubate for a long time, the enzymal activity may decrease. So the right incubation time is very important for optimal or maximum enzymal activity. So time is one affecting factor. Then UV and light radiations. Obviously, UV lights will destroy the enzymes, and destroyed enzymes will denature their proteins. They will get denatured and they will destroy the enzymal activity. So UV lights and radiation will destroy the enzymal activity, and oxidizing reagents and anti-enzymes. So these will contribute to the these are certain factors that contribute to the affecting the normal activity of the enzymes. Okay, so these are the factors affecting the enzymes. So first we understood what is basic, what is an enzyme. Then we understood basic properties of enzymes. Then we understood what are the basic factors affecting the enzymal activity. So this is all about properties and factors of enzymes. So the same thing I kept in another slide, same story. Okay, factors affecting the enzymal activity. Then the next important thing is classification of enzymes, how the enzymes has been classified. Okay, this classification is dependent on the what function does the enzyme did to the substrate, what changes does the enzyme to the substrate. Depending upon the changes the enzyme did to the substrate, these enzymes has been classified. Enzymes has been classified depending upon its activity on the substrate. Okay, now let us see the enzymal activity on substrate and how they are classified. So classification of enzymes. Okay, so enzymes has been classified into 6 categories. Okay, so the 6 categories are, one second, let me keep a small. So the 6 categories are oxidoreductases, transferases, hydrolases, lysases, isomerases, and ligases. So first one is oxidoreductases. So oxidoreductase means, this table is very important, students. I will ask this table in the exams. Okay, so you must be able to plot this table. Okay, classification of enzymes, because we are gonna use all this knowledge. We will incorporate this in the enzyme histopathology or enzyme histochemistry. There we will incorporate these techniques, these functional statuses for visualizing the substrate within the tissue, the enzyme within the tissues. Okay, so yes, this table is very important. So we have oxidases. So oxidases means they will convert to catalyze oxidation reduction reactions. Okay, they will transfer one hydrogen and one oxygen atom or electrons from one substance to another. So here we have AH plus B. Okay, now A will be converted to BH. So from AH, it has an enzyme converted to B. Okay, A's hydrogen ion has been transferred to the B product. Okay, B is product. So this is a reduction reaction and this is oxidation reaction. So here A got oxidized with oxygen. So this is what I got some internet issue, students. That's why I reconnected with my mobile. Okay, so where are we? We are discussing about classification of enzymes. Okay, so coming to the classification, as I stated, we have reductases. Okay, so reductases will oxidoreductases. Okay, either they will oxidize the substrate or they will reduce one hydrogen from the substrate. So this is how oxidoreductases will work. Okay, so the best example of oxidoreductase enzymes are dehydrogenases and oxidases. So dehydrogenase and oxidase is an enzyme that participates in oxidoreductase reactions, where they will transfer either oxygen to the product, to the substrate, or they will transfer one hydrogen. They will remove one oxygen from the substrate. So these are called oxidoreductase enzymes. Then transferases. So transferase enzymes will transfer one molecule from substrate to another molecule. That is the function of the transferases. A few examples are transaminases and kinases. So here A has a B with it, and C is another substrate. So now the B has been converted to C. Okay, so this is how the transferases will work. So in the presence of this enzyme, the substance has been transferred. The B has been transferred from A to C. So this is how the transferases work. So transfer of functional group from one substance to another group, maybe methyl group, acetyl amino or phosphate group is the final primary function of transferases. And the example enzymes are transaminases and kinases. Okay. Then third one is hydrolysis. So hydrolysis, as the name states, they will hydrolyze they will add water to the substrates. Okay, they will add water to the substrate so that the substrate will undergo some other product. The substrate will convert to a different product upon dissolving in water. So best examples of hydrolysis are lipases, amylases, and peptidases. So now, say for example, lipids are water insoluble. So now we, in the presence of this lipase, the lipids will soluble in the water. Okay, the lipase will give one water molecule to the lipid. Okay, this is the primary function of hydrolysis. Okay, examples are lipase, amylase, peptidases. So how hydrolysis work? Formation of two products from a substrate by hydrolysis. So AB plus water. So now it has been converted to A alcohol plus B hydrogen. Okay, so this is how a hydrolysis reactions will work. Then lyases. Okay, so lyases means these are non-hydrolytic addition or removal of groups from substrates having CC, CN or CO or CS bonds. Okay, so these bonds will be removed. Okay, the ketone, carboxyl or nitrogen bonds will be removed by this lyases. Okay, so one best example of this lyase is decarboxylases. Okay, then isomerases. Okay, isomerases will make isomers of the mirror image or the mirror image of the substrate. They are called as isomerases. So best examples are isomerase and mutase. So isomerases will cause intramolecular rearrangement such as isomerization changes within a single molecule is called as isomerases. AB has been converted to BA. So this is the primary function of the isomerases. And last and final is ligases. Okay, so the role of ligases is the best example is a synthesis. So ligases will join any two molecules by synthesis of new CO, CS, CN or CC bonds with a simultaneous breakdown of ATP. So they will use ATP in the chemical reaction in order to combine any two substrates. So this is the primary function of the ligases. Ligases will convert, will join any two substrate molecules in the presence of ATP to make a product, a combined product molecule. Okay, so whenever ATP is used, ATP 1-phosphate is utilized in the reaction. So it has been converted to ADP, diphosphate. Then the single phosphate has been released. Okay, this is how the ligases will work. And best example is synthase. Okay, so these are the classification of enzymes, their reactions and their reaction formulas and their examples. Okay, so we have total 6 types of enzymes. We have oxidoreductases, transferases, hydrolysases, lyases, isomerases, and ligases. So these are the classification of enzymes. Then, how we are going to use these enzymes in enzyme histochemistry? Okay, what is the role of enzymes in enzyme histochemistry? Before that, a quick revision of basics in enzymes. 1 second. So yeah, so as we stated that enzyme will react with a substrate. So substrate, temporarily it will make enzyme substrate complex, then it will be made into a product. Then a catalyst is something that will enhance the speed of chemical reaction. For example, our enzymes will act as a catalyst. Okay, then substrates something that will get affected by the enzymes. Okay, they are called substrates, and substrates will be converted to products. Then properties of enzymes, they will act as a biocatalyst. They will decrease the input energy so that the rate of reaction will be increased. They will speed up the chemical reactions. All enzymes are proteins. Enzymes won't they won't undergo any chemical reactions when they are participating in a chemical reaction, and this same enzyme can be reutilized for the next substrate. And enzymes are reversible and catalyze a reaction in both the pathways. Okay, then factors affecting enzymal activity include concentration of enzymes, concentration of substrate, concentration of products, temperature, pH, actuators, inhibitors, time, UV radiations, oxidizing reactions, and anti-enzyme products within the reactions. So then we discussed about the classification of enzymes. So enzymes has been broadly classified into 6 types: oxidoreductases, transferases, hydrolysases, lyases, isomerases, and ligases. And they will do the following changes to the substrates Either they will transfer, either they will hydrolyze, either they will lyase, either they will change the position or the isomerization of products, okay, the substrates, or they can even join any two substrates. So these are the basic classification of enzymes, their functions, their chemical reactions. Okay. So how these enzymes will be used in enzyme in histochemistry. So as this name suggests, here we are using enzyme in the tissues with chemical reactions. Okay, enzyme histochemistry. So basic introduction to enzyme histochemistry. So enzymes are catalytic proteins in organisms. They are closely related to functional activity in human body, and biochemical reactions can be happen. This we already know. So it also, our enzyme histochemistry will try to localize the enzymes present within the tissues. So coming to the significance of enzyme histochemistry. So certain enzymes found in different cells and organelles, okay, have certain peculiar activities. Okay, so certain specific enzymes present in certain specific tissues in the body. Okay, now understanding these enzymes within the tissues will give us the functional status of the corresponding tissue or the cells. Okay, even in this, and also the activity of these enzymes within the tissue depends also on the status or the demand of or the circumstances of the tissue. Okay, so sometimes tissue need to work hard, sometimes tissue need not to work hard. Okay, so depending upon its demand, the functional status of enzyme depends. Okay, depending upon the demand, the functional status of the enzymes depends. Okay, so even in the same cell, the activity of same enzyme varies because of its functional states, such as in different development stages. During development stage, the functional activity will be more. During terminal phase, the functional activity will be decreased. So depending upon the phase of the development of the cells or the tissues, the functional status of enzymes will varies. Okay, so the strength of enzyme activity has also been used as one of the symbols to judge cell functions. So the status of functional activity, enzymal activity, will give us a clue of the condition of the cell. Okay, because of this, in enzyme histochemical technology, maintaining the exact locations and ensuring the biological activities of enzymes in samples are very necessary. So simple, cancer cells will use more enzymes compared to the normal cells. Okay, because cancer cells are rapidly proliferating, so they are highly energy appetizers. Okay, consumers. They will utilize so much enzymes to develop for their own cancer development. So yes, understanding the activity, the functional activity of enzymes within the cells will give us many insights, certain clues about either neoplasma development, any cancer developments, or any deterioratory effects can be detected within the tissues with the help of understanding the functional status of the enzymes or the activity of the enzymes. So this is the major significance of enzyme histochemistry. So enzyme histochemistry will give us a deeper insights into the tissues and their enzymal activities. Okay, so this is the significance of enzyme histochemistry. So then in tomorrow's lecture, students, we will discuss about various types of enzyme histochemistry reactions. Okay, so usually enzyme histochemistry reactions are kind of method in which a series of reactions are involved. In the reaction process, enzymes in the tissue cell act on their substrates with the production of reaction products under certain conditions, and then the captured agent combines with reactive products, forming colored sediments in light microscope or high electron density products in electron microscope, the visible properties of which can be directly visualized. Okay, very simple. So in enzyme histochemistry, we will take a substrate. To the substrate, we will add an enzyme. So it will make a primary reactive product. Okay, this primary reaction product can be stated as a enzyme product conjugate, enzyme substrate complex. Okay, so this is called PRP. Okay, primary reactive product. Now to this primary reactive product, we will add a capture color. Okay, we will add certain colors, so that colors will develop. This product will be visualized in the microscope. So we will add some capture substances. These capture substances will develop color to the color to this product. That product can be visualized in various types of microscopes. They can be visualized in electron microscopes, light microscopes, even specialized polarizing, phase contrast, or even other microscopes can be utilized for the visualization of the product, okay, within the tissues. So this is the idea of enzyme histochemistry. So we are incorporating enzymes, and we are incorporating a capture coloring substance that will develop colors. So this is the basic idea of enzyme histochemistry. So as I stated, a brief introduction again. So enzyme histochemistry serves as a link between biochemistry and morphology. It is based on the metabolization of substrate provided to a tissue enzyme in its orthotopic localization. Visualization is accomplished with an insoluble dye product. It is a sensitive dynamic technique that mirrors even early metabolic imbalance of any pathological tissue lesions, combined with the advantage of histophotographic enzyme localization. With the advent of immunohistochemistry and DNA-oriented molecular pathology techniques, the potential enzyme histochemistry has been okay. Nowadays, we are not using this enzyme histochemistry because we got another advanced methods like immunohistochemistry and DNA-oriented molecular pathology has taken. They are dominating in the fields of this histopathology. So nowadays, our enzyme histochemistry is not used much. But the best advantage of this enzyme histochemistry is its low cost. Enzyme histochemical techniques are really cheap and effective compared to immunohistochemistry techniques or molecular pathology techniques. Okay, so enzyme histochemistry has its own significance where this will be really helpful in simple lab setups. Okay, and the cost of the test will be less. So in cost-effective regions, we can opt for this enzyme histochemical techniques in order to visualize the specific components within the tissue. Okay, so this is the major advantage of enzyme histochemistry. So the same story. So enzyme histochemistry principle. Enzyme histochemistry combines the biochemical activity of enzyme with the information about its localization. The basics of enzyme histochemistry has been described extensively in 1970s itself. Okay, so the general reaction of enzyme histochemistry is reaction between enzyme and substrate produce a primary reactive product using a histochemical reaction involving, for example, chemicals like diazoamium salts produce a stained precipitate product. The product can be represented like this. So we got a substrate acted with an enzyme, made PRP, primary reactive product. Now this primary reactive product will be subjected to a capture molecule, which is the diazoamium salt. This diazoamium salt will develop color. This color can be visualized under the microscope. So this is enough for today's lecture, students. In tomorrow, we will go further and we will see individual chemical reactions, and at last we will see demonstration of various types of enzymes within the tissues. So in today's lecture, we discussed about the basics of enzymes, their properties. Then we discussed about the catalytic properties, their factors affecting enzymal activity, and we also discussed about various classifications of enzymes. Okay. Then we also started a brief introduction on introduction to enzyme histochemistry, significance of enzyme histochemistry, and the basic principle and overview of enzyme histochemistry. So in tomorrow's lecture, we will discuss in detail about further important topics in enzyme histochemistry. Thank you very much, students.